Hello, everyone. My name is Chris Mathis, and I'm Chief Commercial Officer at AnaBios. It's my pleasure to welcome you to this AnaBios-sponsored webinar today entitled Evolving Concepts of Atrial Fibrillation Mechanisms, Relevance to Therapeutic Innovation. So before I introduce the speakers, I would like to tell you a little bit about AnaBios. Some of you know us, some of you may not. AnaBios is a unique contract research organization and biotech company located in San Diego. We recover human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. So we have essentially redefined first in human. In other words, working with AnaBios is as close to get to a clinical trial without actually doing a clinical trial. So it's my pleasure to introduce first my colleague, Dr. Najah Abi-Gerges. He's the Vice President of Research and Development at AnaBios, and he's been with the company for about 5 years. He received his PhD in cardiac physiology from Paris University in France. And prior to joining AnaBios, Dr. Abi-Gerges was involved in drug discovery programs at AstraZeneca and made substantial contributions to drug approvals, for example, Tigrosia. He has been involved in research across several areas of cardiac physiology and pharmacology, resulting in over 40 published peer-reviewed articles and novel paradigms to advance cardiovascular translational science. Dr. Abi-Gerges is the editor of the Journal of Pharmacology and Toxicological Methods and is also a reviewer for more than 20 peer-reviewed scientific journals and review judge panel for various science institutes and foundations. And in the past 6-7 years, he's been the co-chair of the Ion Channel Group for the CEPA initiative. So the title of his presentation today is Human Heart Atrial Models for the Development of Novel Anti-Atrial Fibrillation Drugs. Najah, take it away.
Thank you, Chris, for the introduction. Hello, everyone, and welcome to this webinar. For the next 10 minutes, I will give you an overview about the human primary cardiac models that we have developed at AnaBios, and they can be used to support cardiac drug discovery programs, including atrial fibrillation program. At AnaBios, we developed novel method to recover and ship human organs while the highest quality and viability are ensured. These human organs can be used to profile drug activity on the live human cells and tissue, and also they can be used to conduct omic analysis. One of the human organs we recover is the entire human heart. We have worked on over 1,400 human hearts today, and we have a consistent recovery that goes on a weekly basis. As you can see in this slide, we developed tissue-based model and cell-based model. The tissue-based model can be used to support late preclinical development, while the cell-based model can be used to support early drug discovery. And you can see again from this slide that we have a variety of different models that they can be used to assess the potential of novel drugs to modulate ion channel, to modulate action potential, modulate calcium dynamics, and also to modulate contractility, chronotropy, and also we have model to assess their potential to induce vasoconstriction and vasodilation. In addition to this, to recovering a normal human heart, also we can recover hearts with disease like atrial fibrillation, hypertrophic cardiomyopathy, MI, and heart failure. All these models, can be used to support cardiac drug discovery. The next slide shows a potential discovery strategies that can be applied to support AFib programs, where human data can now be used in decision making from early drug discovery phases all the way to nomination of clinical candidate drug and all the way to clinical trials. Again, as you can see in the strategy, we have different in vitro or different cell and tissue-based models that they can be positioned to support a program. But in summary, the models can be used to establish or find novel targets. They can be used to find lead compounds and then to define mechanism of action. And all this data can be used basically to confirm a human atrial efficacy of novel drugs. While we are conducting this atrial efficacy data, you can see at the same time we can conduct a study to confirm the atrial selectivity of the novel drugs, while we confirming that they don't affect ventricular function and they are not associated with any potential to cause proarrhythmia. What I will do with the next two slides, I will show you a case study, how we supported a discovery atrial fibrillation program in confirming the potential of a drug X to be an anti-AF drug to treat human AFib. Basically, the discovery team has some data suggesting a possible potential for drug X to be an anti-AF drug. So what we did, we used our human primary cardiac models to confirm the human efficacy of drug X. So as shown in this slide, data indicate that the drug X prolonged action potential in atrial myocyte isolated from the heart of patients with atrial fibrillation. And also the data indicate that the drug X prevented abnormal contractility and arrhythmia in atrial trabecula, again, isolated from the heart of patients with atrial fibrillation. Taking together, this data, the data indicates that the models can provide preclinical human efficacy proof of concept data for drug X to be a very credible anti-AF drug. Next, what we did, we wanted to confirm that drug X has a good cardiac safety. So we used also, again, our models to confirm this. As it can be seen on this slide, drug X was found not to modulate action potential in atrial myocyte and also ventricular action potential in the model of ventricular trabecula. Both atrial myocyte and the trabecula were isolated from normal heart. Next, we confirmed again that drug X had no ability to modulate contractility. And we confirmed this in the human primary cardiomyocyte model, where we measure sarcomere shortening, and also we confirmed this in the ventricular contraction model, where we measure force of contraction. In addition to this, the ventricular action potential data and the data we can derive from the ventricular myocyte model, like after contraction, contraction failure. All this data indicated that drug X has no potential to cause proarrhythmia. In recent two publications in Frontiers in Physiology and Scientific paper, we demonstrated that after contraction and contraction failure events are good predictors of proarrhythmia. Taking together this human cardiac efficacy and safety data confirms that the drug X is really a very credible anti-AF drug. This data played a critical role in helping the discovery team deciding to take drug X to IND application in 2021. The next three slides will give you an idea about the role of late sodium current in the human heart. Recently, we have conducted a big study characterizing the role of late sodium current in the human ventricle and the human atria. Unfortunately, there is no time today to present the entire data set, but we will show you some data on how our models can be used to better understand the role of late sodium current in the human atria and confirm its potential to treat atrial fibrillation. In this slide, you can see in atrial human myocyte, application of ATX2, an enhancer of late sodium current, was used to cause arrhythmia. You can see that ATX2 application increased the contractility and also caused the incidence of after contraction and contraction failure. Application of GS967, an inhibitor of late sodium current, as you can see, reversed the increase in contractility and also significantly reduced the incidence of arrhythmia. The take-home message from this data is that late sodium current inhibition restores normal atrial myocyte contractility and prevents arrhythmia. Next, we wanted to confirm that this atrial myocyte data will translate well to atrial tissue, i.e. does data translate from a cellular model to a multicellular preparation. Again, in this slide, we used ATX2 to increase, ATX2 caused an increase of contractility or force of contraction in atrial and ventricular trabecula isolated from normal heart. In addition to increasing contractility, ATX2 caused after contraction incidence, and also in the atrial trabecula caused enhanced automaticity. Again, inhibiting late sodium current with the GS967, as you can see, we reversed the increase in contractility, we restored the normal contractility, and of course, we prevented incidence of atrial arrhythmia. Next, we wanted to get a better idea about the role of late sodium current in atrial fibrillation. As shown in this slide, application of ATX2 again caused increase in contractility and incidence of after contraction. And this was seen in atrial and ventricular trabecula that were dissected and isolated from the heart of a patient with atrial fibrillation. Again, inhibiting late sodium current with the GS967, you can see that we restored normal contractility, and the late sodium current inhibition completely blocked the incidence of arrhythmia in atrial fibrillation condition. Taking into consideration this data confirm again the credible potential of late sodium inhibitors to treat atrial fibrillation in the human heart. This slide is to conclude on my quick presentation today that other human heart models can be used to support atrial fibrillation drug discovery program. They can be used, or they can permit generation of a human efficacy proof of concept data at the preclinical stages. They can help to confirm atrial selectivity of novel anti-AFib drugs, and also they can help to ensure that novel anti-AFib drugs lack pro-arrhythmic potential. Thank you for your attention, and now I will hand over to Chris so he can introduce Dr. Stanley Nattel. Thank you.
Thank you, Najah, for that excellent presentation. And just a reminder, if you have questions for both of our speakers today, we will have a Q&A session at the end. And you can write your question in the lower bottom panel that says questions, and we will get to those. We will not be doing oral questions today for the sake of time, so please get your questions in to the right, even during the presentations. So it's really my pleasure to introduce our keynote speaker from the University of Montreal, esteemed scientist and physician, Dr. Stanley Nattel. He received his MD from McGill University in 1974, undertook internal medicine and clinical pharmacology training at McGill from 74 to 78, then obtained clinical and basic research training at Indiana University and University of Pennsylvania before returning to McGill in 1981. In 1987, Dr. Nattel transferred to the University of Montreal and Montreal Heart Institute, where he directed the research center between 1990 and 2004. He is presently Paul David Chair in Cardiovascular Electrophysiology at the University of Montreal and Director of the Electrophysiology Research Program at the Montreal Heart Institute. He is Editor-in-Chief of the Canadian Journal of Cardiology, Associate Editor of Cardiovascular Research, Rhythm Disorders Section Editor in JACC, and is on the editorial board of a number of other journals, including Cardiovascular Research, Circulation Research, Circulation Arrhythmia, and Electrophysiology Drugs, JACC, Clinical Electrophysiology, and many others. His research focuses on clinically relevant mechanisms of cardiac bioelectricity, particularly atrial fibrillation, proarrhythmia, cardiac remodeling, ion channel molecular physiology, and mechanisms of drag action. His lab uses a wide range of molecular, cellular, whole animal, and mathematical modeling methods to study clinically relevant basic mechanisms and identify novel therapeutic agents. He has supervised over 170 research trainees, published over 675 papers in peer-reviewed journals, and has an H-index of 136. He's a fellow of the Royal Society of Canada, Academy of Science, the Royal College of Physicians of Canada, the American College of Cardiology, and the Heart Rhythm Society. So it's my pleasure to introduce Dr. Nattel today, and the presentation is titled Evolving Concepts of Atrial Fibrillation Mechanisms, Relevance to Therapeutic Innovation. Dr. Nattel, please take it away.
Okay, so thank you very much, Gary and Najah, for giving me this opportunity to talk about a subject that I think is very interesting, which is the evolving concepts of AF mechanisms and their relationship to therapeutic innovation. This is an outline of my presentation. I'll be talking first about conceptual models of AF. What are they and what is the practical difference among them? Then I'll go through a discussion of electrically active drugs for AF termination and prevention and discuss the challenges and whether such agents can be improved. And finally, I want to move to a more future-looking subject and talk about prevention of the AF substrate and the role of inflammation. So first of all, conceptual models of AF, what are they and what is the practical difference? So the classical model that most of us were taught in medical school or physiology courses was the multiple circuit reentry model, which was initially put forward by Gary and Mines in 1914, then adapted by Gordon Moe with his multiple wavelet hypothesis, and really forms the basis of a lot of teaching and thinking about AF. However, recent work over the past 30 years or so has revived very old ideas about AF in terms of the arrhythmia potentially being induced and maintained by local foci firing extremely rapidly, or via local rapid reentry circuits in the pulmonary vein that fire so fast that the rest of the atria can't keep up and fibrillatory conduction results. Over the past several years, a few additional models have been introduced and have received experimental support and varying clinical support. So one idea is that of stable reentrant rotors that maintain fibrillation via fibrillatory activity and are potentially interesting sites for ablation. This idea was first put forward by Pepe Jalife, and then Sanjeev Narayan provided clinical data to support it, which I'll show you. Other thinkers have more recently showed evidence that, in fact, stable rotors are uncommon, but more typically one sees unstable rotors anchored in fibrotic zones. And finally, Moritz Alessi has challenged over the past 10 years the notion that atrial fibrillation is maintained by epicardial endocardial interaction and maybe not classical reentry or rotors as we think of them. So the idea of stable reentrant rotors, as I mentioned, was put forward first by Pepe Jalife, and then Sanjeev Narayan developed his mapping system, which uses basket mapping and sophisticated computer analysis to identify rotors in the left or right atrium. And Sanjeev initially reported a very high prevalence of stable rotors. He called this technique FIRM for ablation of sources and rotors, and showed that local ablation at an identified rotor site could terminate AF in many patients. And he identified long-lasting rotors lasting over 10 minutes in all patients. He published a controlled trial showing a very high level of freedom from AF in patients that were treated with the FIRM technique, and in patients who were FIRM blind, so they underwent the same interventions, but they were not given the FIRM data, had a much higher recurrence rate, and this was highly significant and published in JACC. So obviously, the success of applying the concept of rotor ablation depends on the existence and ability to identify spatially stable rotors. After the initial enthusiasm about Sanjeev's work, a large number of other investigators tried the same methods, and there was great variability in the success rate of different methods to apply this rotor ablation technique to terminate and prevent AF. So after Sanjeev Narayan's initial studies and those of others, a variety of later studies had much lower success rate, and the overall success rate in paroxysmal AF, non-paroxysmal AF was reported to be 59%. And this is a study by Atul Verma in the New England Journal of Medicine. He didn't use rotor ablation, but he showed that pulmonary vein isolation was comparable to more aggressive ablation techniques in patients with AF. And if you take the aggregate rotor ablation results, they really sit right on what Atul found for pulmonary vein isolation alone. So sustained stable atrial rotors maintaining AF are absent in many patients, or we're not detecting them adequately in order to understand why this rotor ablation technique doesn't work so well. Michel Saguer applied ECGI mapping in combination with MRI imaging of fibrosis, looking at late gadolinium enhancement, in order to look at rotors and at their location in relationship to atrial structure. So this is an example of a rotor identified by ECGI. ECGI is a torso imaging method where electrical activity is recorded at many points on the torso, and this is transferred into a reverse analysis, the so-called inverse solution of electrical activity at the level of the heart. And as you can see in this slide, the phase singularities corresponding to the center of a reentrant rotor could be mapped in AF patients, and they tended to cluster at the edge of fibrotic zones. So here, fibrotic zones are identified in white. The phase singularity trajectories are these spaghetti-like lines, and you can see that the phase singularities are sometimes in one specific area. More often they're in multiple areas and meander around, but they tend to cluster at the edge of fibrotic tissue. Very much unlike Sanjeev Narayan's data, the mean number of rotations maintained by each rotor was very small. So these rotors that lasted about 200 milliseconds each, they had a mode of about 2.8 rotations, so they only lasted under a second in most cases. So each rotor is short-lived, lasting an average of 2 to 4.2 rotations, and these data suggest that rotors do occur, but are evanescent and only weakly anchored by functional barriers. So these data were interpreted as supporting the notion of unstable rotors that were anchored in fibrotic zones. Moritz Alessi's notion of endocardial-epicardial interaction came out of detailed mapping studies in the operating room, and this is an example of his mapping data in acute AF and persistent AF, and you see that in persistent AF, the activity is very much more complex, and it looks as though there's longitudinal dissociation between fiber bundles because you have independent areas of activity. And the same year, de Groot et al. Reported from Alessi's group reported evidence of endocardial to epicardial breakthrough, providing the appearance of sources, of focal sources, and presumably promoting AF maintenance via a multiplication phenomenon where the breakthroughs resulted in multiple activation regions and propagation of the impulse. So this epicardial endocardial interaction was identified as a mechanism for the multiplication of active wavefronts and AF maintenance. A study that to some extent pulled some of these ideas together was published by Hansen et al. In the European Heart Journal. They used very sophisticated mapping techniques with two highly sophisticated optical mapping systems on human cardiac preparations. They noted evidence of stable rotors that used a transmural port that had a critical transmural portion and went from the epicardium to the endocardium to maintain AF. And here's an example of AF with a cycle length of 64 milliseconds, where the transmural reentrant circuit was targeted by radiofrequency ablation that produced a slowing of the activity and regularization into an atrial tachycardia, and then following an additional targeted radiofrequency lesion, this heart returned to normal rhythm. So these data support and unify several of these ideas by suggesting that epicardial endocardial interaction may be due to transmural reentry circuits that use areas of fibrosis in the myocardial wall as anchors for local reentry circuits. In Hansen's study, these circuits could be stable, but would not be obvious at the surface if you recorded only from the epicardial surface alone because of the involvement of the endocardium and transmural sites. And they could appear as AF maintaining foci through breakthroughs to the epicardium. So this highly sophisticated mapping points to AF maintaining reentry paths involving endo-epi interaction with transmural conduction. Such activity could provide stable reentry circuits that might be manifest as focal activity upon strictly epicardial recording. And these circuits are amenable to termination by ablation. However, and this is an important limitation that needs to be recognized, these patients did not have spontaneous AF. These were failing hearts that were removed at the time of transplantation, and they used large doses of adenosine to stabilize the arrhythmia. So whether more physiological conditions would produce AF via unstable rotors, as seen by Eyseguer et al., remains an open question. So in summary, a variety of models have been put forward to understand AF, but the available data are unclear. The clinical implications of the underlying mechanisms, especially for persistent AF, are great. However, they remain poorly understood, and the challenges to improve translation require intense further work to overcome. Now I'm going to talk about electrically active drugs for AF termination and prevention, discussing some of the challenges and what's needed for improvement. So this is sort of the classical notion developed by Moritz Alessi in the 1990s of the determinants of atrial fibrillation. Moritz described the wavelength as the determinant of functional reentrant circuit size. The wavelength is the minimal path for reentry and the size of functional reentry circuits, and it's the product of refractory period and conduction velocity. So in normal atria with normal wavelength, AF is not sustained. In normal atria with a short wavelength, you can have multiple simultaneous reentry circuits and AF is sustained. So to stop AF, presumably all you need to do is to increase the wavelength by increasing action potential duration. On the other hand, as we all know, the risks of action potential duration prolongation are well recognized. IKR blockade reduces an important repolarizing current and prolongs the atrial action potential, producing an important anti-AF effect. However, IKR is also strongly expressed in the ventricle, and when you block IKR in the ventricle, you get ventricular APD prolongation with a risk of torsade de pointe and life-threatening proarrhythmia. So the notion of potassium channel blockers with atrial selectivity was put forward, and a variety of ion currents were identified that were atrial selective and could potentially act as targets. One of these we discovered in the early 2000s, the ultra-rapid delayed rectifier current, which is expressed only in the atrium. These are ion current recordings from human cardiac cells, and we identified IKUR by its typical very high level of sensitivity to 4-aminopyridine. So 50 micromolar 4-aminopyridine blocks about half of the IKUR in the human atrium, revealing a very clear, very rapidly activating current. And in the human ventricle, you don't see any of this. And this is a study However, there are some important translational challenges to RKUR as a target. This is from a modeling study that Mark Courtemanche and my group performed in 1999, and he showed that if you block IKUR at least in the standard Courtemanche action potential model of the human atrium that we developed, if you do it in normal atrium and block only IKUR, you see no net change in action potential duration, because even though early repolarization is slowed and you get this enlarged plateau, IKR becomes activated, causes acceleration of late repolarization, and there's no net APD prolongation. On the other hand, in AF action potentials, because of the remodeling, the underlying action potential is triangular. There's much less IKR activation, and you do see some prolongation, but relatively modest. So the degree of action potential prolongation with IKUR blockers may be limited, especially in the absence of remodeling. This is another potential problem with IKUR as a target. This is a study reported by Tim Olson, in which they looked at a family with KV1.5 mutations. These caused a loss of function in IKUR, and at least in an in vivo experimental model was able to cause triggered activity and proarrhythmia. So they suggested that at least in some cases, loss of function in IKUR may actually promote AF. So while theoretically very attractive, the value of targeting IKUR remains uncertain. Another target that was identified is IKCH. These are recordings of ion currents from dog atrium. These are from a control dog. This is from a cell in a dog exposed to AF-related atrial tachycardia remodeling. And if you just look at the current sensitive to tertiapin, which is a highly selective IKCH blocker, you see that there's a component, even in the absence of cholinergic agonists, of ion current that is sensitive to tertiapin in control, and this gets increased with atrial tachycardia remodeling. So this is a constitutive IKCH current because it doesn't require the presence of a cholinergic agonist, and it increases with AF-related remodeling. And Tae-Jun Cha in my lab was able to show that there's enhanced action potential prolongation in atrial tachycardia remodeled atria, and that tertiapin Q could terminate atrial tachyarrhythmias induced in canine atrial preparations from dogs that had undergone AF-related remodeling. So this is very nice and suggests that blocking IKCH might be an interesting target in humans. In dogs, we found that it was able to convert AF in atrial tachycardia remodeled animals. However, in a human study, atrial refractory period was not altered by a candidate IKCH blocker in patients with a history of atrial flutter. Now, the big question is whether the approach doesn't work in man, or whether IKCH is only significant in the presence of atrial remodeling, and you would only, drugs with IKCH blocking capacity would only be of value in patients with already electrically remodeled atrial tissue. Of course, if that were the case, they wouldn't be particularly interesting in preventing AF in individuals who are in sinus rhythm and not in AF. So what about sodium channel blockade in AF? So the expected effect of sodium channel blockade is to decrease conduction velocity and decrease the wavelength, and theoretically, at least according to the leading circle model, you should take normal atria and make them more AF prone. But clinically, sodium channel blockers are effective in AF, and you can see this is the efficacy percent cardioversion with a variety of sodium channel blockers compared to placebo, and you can see that propafenone is highly effective. Quinidine, amiodarone, sotalol are much less effective. These drugs are not as effective sodium channel blockers as propafenone. So in order to try to understand why sodium channel blockers might work in AF, we looked at the relationship between the intensity of sodium current blockade and AF termination in a realistic two-dimensional mathematical model of AF in human atrium. And we did see an efficacy for AF termination that was related to the extent of sodium channel blockade in the model, so that's good. And then explained or understood why sodium channel blockers are working based on the rotor model of AF. So as I mentioned, the classical model was the leading circuit that depends, where reentry depends on the balance between refractoriness and conduction. If you slow conduction, you should decrease the wavelength, make the circuit smaller, and promote AF. But an alternative model is that of the spiral wave or rotor, which may be stable or unstable, as I showed you before. And a key concept in the rotor model is the critical role of curvature in the spiral. So to try to simplify this, because it could be a very long discussion, a rotor activity is maintained by an active wavefront that has an excitable but not excited core. And the key to understanding this is, as I mentioned, the role of curvature. So at the periphery of the spiral, you can see that the wavefront is relatively flat. And each cell at the periphery will be activating a similar number of cells beyond it. And so you don't need a very strong source to maintain activity. So where the wavefront is not curved, the zone being excited is equal to the source, and you have a good source sink match. However, if you look towards the center of the leading wavefront, there you have strong curvature, and each cell needs to activate multiple cells beyond it. You can see that the source zone is relatively small compared to the zone that has to be excited. So the zone being excited is much larger than the source, and you have a large source sink mismatch, which at the core tip results in lack of excitation, and that's what produces this core zone, which is excitable but not excited. And at the center of this zone is the phase singularity, which is equivalent. This concept of a rotor is, by the way, very common in biology and in science beyond biology. And for example, a tornado or a hurricane is an example of a rotor, and the eye of a hurricane is essentially the same thing as the core of a cardiac reentrant rotor. So these rotors differ from the leading circle in that the core is not kept refractory by centripetal waves, unlike in the leading circle. There's no excitable gap, which you see in the leading circle, but not in the rotor. And maintenance and properties depend critically on the excitability, which is determined by the propagation strength and refractoriness that determines the size and curvature of the rotor, not on a balance between conduction velocity and refractory period. So if we look at the functional determinants of a rotor, you have the activation wavefront, the core with the phase singularity at its center. You have depolarized tissue in the active propagating wave, which is with a refractory zone determined by the refractory period. And this produces the activation wavefront produces an excitatory current, and this current that causes excitation then interacts with the tail of the refractory zone, and that zone has to be able, the zone behind it has to be able to be excited in order to maintain the reentry. So perpetuation depends on the ability of the wavefront to continuously activate tissue, which depends on the strength of the excitatory source and the magnitude of the current sink. So bringing us back to sodium current, a smaller sodium current decreases the source strength, decreases the ability of this activation wavefront to constantly excite the tissue in front of it. So decreased excitatory current causes reentry to terminate when the current at the excitatory wavefront becomes small and destabilizes the rotor. And that explains the efficacy of sodium current blockade in AF. Now this can be exploited to the concept of the rotor and the determinants of reentry can be exploited along with the state-dependent sodium channel block in order to produce more AF selective sodium channel blockers. So if we look at block indicated by red and blue and unblock indicated by blue, during AF, the atrial rate is very rapid. In sinus rhythm, it's much slower. So in sinus rhythm, a sodium channel blocker will interact during the depolarized phases of the action potential and then unblock during the diastolic phase. During AF at its very rapid rate, there's more interaction, the interaction during the excited phase is similar, but the diastolic portion is much shortened, and that reduces the unblock and increases the degree of sodium channel block. So it's possible to play with the activation and inactivation rate constants, and these are the binding rate constants, but we looked at both binding and unbinding rate constants in order to predict sodium channel blockers that would be highly selective for terminating AF with very little action during sinus rhythm. So we found that the inactivated state binding is the main determinant of AF selectivity, and we were able to identify a zone in which drugs had some degree of atrial selectivity. So this color chart indicates the degree of selectivity for atrium versus ventricle, and you can see it goes up to about a 2-to-1 ratio in this zone of combination of binding rate constants. So the atrial selectivity for the atrial action potential versus ventricular in sinus rhythm is relatively small, although some can be achieved. But the AF selectivity is much greater because of the reduction in unbinding during the short diastolic period. And it was possible to identify a zone of binding and unbinding rate constants where the efficacy was very high, getting 100% termination, and a proarrhythmia index that we identified by looking at atrial versus ventricular models of arrhythmia was relatively low in this region. So this approach identifies optimized sodium channel blocking drug pharmacodynamics for effective AF termination with minimized proarrhythmia risk, and could be developed by industry to find more rational sodium channel blockers. We then asked whether the addition of moderate potassium channel blockade could be used to enhance the selectivity, the AF selectivity of blocking drugs. So if we look at AF and the blocking and unblocking portions of the action potential, and then model AF with a potassium channel blocker, the depolarized potential increases and the diastolic, the depolarized portion increases relative to the diastolic repolarized portion, and presumably you would have much more ability to get much more AF selectivity with sodium channel block. And so when we combined a 50% IKUR block and then looked at the optimally AF selective sodium channel blockers, we were able to achieve an AF selectivity of a ratio of 97, so that's 97-fold greater sodium channel blockade on atrial tissue during AF versus ventricular tissue in sinus rhythm. So this is the AF selectivity with sodium, with IKR block at 50%. Without IKR block, we were able to achieve AF selectivity, but to a lesser extent. So these observations, in addition to explaining why drugs like amiodarone might be very effective by blocking both potassium and sodium channels, suggest that it may be possible to exploit this for new drugs with selected degrees of blockade of both. And here we looked at IKUR block as an atrial selective target in combination with sodium channel block, and at least in our mathematical model, we could get tremendous AF selectivity by combining partial IKUR block with the sodium channel blocker. So this general approach may eventually allow for the development of designer AF suppressant drugs, possibly exploiting the synergy between atrial-specific IKUR blockers and sodium channel blockers. Very important to recognize all this modeling work was done with models of human atrial tissue and ventricular tissue, and is critical to recognize the species-specific differences in studying such compounds. So in summary, improved antiarrhythmic drug therapy will require more selective drugs. Atrial selective potassium channel blockers are one possibility, but they have to be tested on human models because predictions from animal models may not work out. AF selective sodium channel blockers are another potential approach, and combining atrial selective potassium channel block with AF selective sodium channel blockers may provide optimal AF selectivity. Now, I want to finish it in the last 10 or 15 minutes by talking about prevention of the AF substrate and the role of inflammation. So before I go into the specific role of inflammation, a lot of my discussion till here has been only about the electrical aspects of AF. But over the past 20 or 30 years, it's become obvious that with remodeling of the atrium, as happens with cardiac disease, risk factors, aging, and with AF itself, if it's maintained long enough, patients develop fibrosis and other changes in the atria that stabilize long-standing AF and make it very resistant to therapy, whether drug therapy or ablation therapy. So we've been working for a long time on trying to identify targetable intermediates that connect risk factors to the development of the AF substrate to try to identify targets that might be clinically usable to prevent the development of the substrate for AF rather than simply targeting the final electrical product. So this is a study from Mina Chung and David Van Wagener, was published in Circulation in 2001, and they noted that patients with AF have an increased C-reactive protein level. And furthermore, so this seemed to be the case in different forms of AF, but there was a progressive increase in the CRP level, which is an indicator of inflammation, a very non-specific indicator, but nevertheless is an indicator of inflammation. And you could see that there's a graded increase in CRP level with paroxysmal and then even more with persistent AF. So I want to present the results here of a couple of studies which I find intriguing, which suggest that it may be possible to target, first of all, they suggest that cardiomyocyte inflammatory signaling may be very important and that it may be possible to target atrial inflammation to prevent fibrosis development and development of the substrate for AF. So this is a study from Nali and Zander Verenz and Dobromir Dobrev that I had the fortune to be involved in. And what we found was that indicators of NLRP3 inflammasome activation were increased in tissue from dogs and mice, dog and mouse models of AF, and also in AF patients. Now, without going into any detail in the NLRP3 inflammasome system, this is a system that's critical in inflammation. It's expressed in inflammatory cells and causes release of cytokines and local inflammation. But these data show that cardiac inflammatory signal is enhanced in AF patients and AF animal models, and further work that I don't have time to present provides the intriguing observation that the inflammatory signaling is strongly expressed in cardiomyocytes. So cardiomyocytes seem to have the machinery for inflammatory signaling that was previously thought to be restricted to inflammatory tissue, inflammatory cells, to white cells. So Na developed a mouse model with cardiomyocyte restricted activation of NLRP3. And she found that these mice had a higher incidence of atrial premature complexes and a greater inducibility of atrial arrhythmia. So here you see a brief protocol to induce AF, which did not induce arrhythmias in the control animal, induced AF in the cardiomyocyte knock-in NLRP3 animals, and then an organic blocker of NLRP3 suppressed arrhythmia induction. And here are the mean data for incidence of inducible AF, which increased with the knock-in of the NLRP3 activation system and were suppressed by this organic blocker. So enhanced cardiomyocyte inflammatory signaling is sufficient to create AF susceptibility and atrial ectopy in the mouse. They then asked whether knocking out this system in cardiomyocytes might be enough to prevent AF in a spontaneous AF model that didn't involve NLRP3 activation genetically, but had AF on a different basis. So they used the CREM mice, which is a well-established mouse model for spontaneous atrial fibrillation, and they noticed that procaspase-1 and caspase-1 P20, which are indicators of NLRP3 inflammasome signaling, were enhanced in the CREM transgenic mice and suppressed by knockdown of NLRP3 in these mice, actually knockout. So these were double transgenics that had the CREM expression as well as NLRP3 knockout. And you can see in the right panel that procaspase-1 is increased in the CREM transgenic suppressed with NLRP3 knockout, that the incidence of spontaneous AF is greatly enhanced in the CREM transgenic and suppressed in the double mutant with the NLRP3 knockout, and that atrial remodeling in terms of left atrial size on echo is increased in the CREM transgenic mouse and normalized by knockout of NLRP3. So suppressing cardiomyocyte inflammatory signaling prevents atrial remodeling and AF in a mouse model of spontaneous AF. And then one can ask the question, how can one practically interfere with inflammatory signaling, which is such a critical component of our human response to infection, disease, and healing? So there's a concept which has been introduced in the last few years, which examines a novel biochemical system that contributes to inflammation resolution, preventing chronicization of inflammation. So the idea here is that when inflammation initially occurs, it briskly activates inflammatory components, deals with the primary stimulus or stress factor that causes the inflammation, and then normally resolves. But in some individuals, there's failure of resolution of inflammation, which results in chronicization of disease. So if we look at the different derivatives of polyunsaturated fatty acids, so we have arachidonic acid, which generates prostaglandins and leukotrienes, which are well known to be pro-inflammatory, but also generates other substances like lipoxins and aspirin-triggered lipoxins that promote resolution of inflammation. EPA generates a resolvin, so-called resolvin molecule that helps in the resolution of inflammation, and docosahexaenoic acid, DHA, generates a series of inflammation resolution promoting molecules. So the background idea to this is that the failure of polyunsaturated fatty acids to have efficacy in dealing with human disease may be due to the fact that these compounds produce both pro-inflammatory components and components that are derivatives that are necessary for the resolution of inflammation. And by using the parent, one creates a balance of effects that may not be effective, but that if one could use only the inflammation resolution promoting moiety, that this might be beneficial. So we tried this in an animal model. We first developed a rat model of right heart disease using monocrotaline to produce pulmonary hypertension. And in these rats, we noticed that there was very strong promotion of AF. This is the percent of induced arrhythmias in monocrotaline treated rats compared to control rats, and prolongation of the P wave indicating slowing of atrial conduction. In fact, there was extensive fibrosis. So these recordings were from in vivo electrophysiologic study with transesophageal stimulation. And we also looked at Langendorf perfused hearts and in situ electrophysiologic studies, and again, saw very clear AF promotion with monocrotaline-induced pulmonary hypertension and right heart disease. And again, you see the percentage of AF induction is much greater, and AF duration is substantially prolonged with pulmonary hypertension. So these monocrotaline treated rats have increased susceptibility and sustainability of AF. And we found evidence of inflammasome activation in these rats. So this is a very simple schematic of the NLRP3 inflammasome system. NLRP3 interacts with ASC and pro-caspase to generate active caspase 1 and caspase 8 that increase membrane permeability to interleukin-1 beta and cause inflammation. And we found that although NLRP, so these are, in these graphs, you see on the vertical axis mRNA expression of a variety of inflammasome components and products, and you see also on the horizontal axis the different conditions we studied. So control right atrium and left atrium, right atrium and left atrium from monocrotaline treated rats, and we also as a control group used MI rats that had left atrial remodeling. So the monocrotaline treated rats had AF based on a right atrial substrate, whereas the MI rats had it based on a left atrial substrate. And you can see that NLRP3 itself was expressed to a greater extent, but not significantly in these rats. But caspase 1 was significantly enhanced, both in the monocrotaline and the MI model. Caspase 8 was significantly enhanced, and interleukin-1b was enhanced in all of them. The error bars were too large in the monocrotaline rats to achieve significance. In the MI rats, it actually achieved statistical significance. So these data suggest that inflammatory signaling gene expression is enhanced in monocrotaline and MI atria. And then we gave Resolvin D1 for the 21-day conditioning period after monocrotaline administration and studied the effects on AF and atrial remodeling. So these are control, this is AF inducibility in control rats and control rats treated with Resolvin D1, no inducible AF, with monocrotaline, substantial increase in AF inducibility, just like the other data that I showed you, and Resolvin D1 substantially reduced AF inducibility. This is right atrial fibrosis with monocrotaline. You can see quite dramatic fibrosis, which was significantly suppressed by Resolvin D1, and there was no clear effect on right atrial size. The effect was really restricted apparently to an anti-inflammatory action that reduced atrial fibrosis. We see the same kind of phenomenon in the left atrium in the monocrotaline treated rats, increased left atrial fibrosis, suppressed by Resolvin D1, and again, no change in left atrial size. So Resolvin D1 attenuates in vivo AF vulnerability and atrial fibrotic remodeling in monocrotaline rats, and could be an interesting anti-inflammatory target, as Resolvin appears to be fairly non-toxic in animal studies. So in summary, inflammatory signaling is a potentially important contributor to AF substrate development. It's activated in humans, and activation in animal models is sufficient to produce fibrosis and also electrical remodeling, which I didn't have time to show you. Inflammatory signaling can occur in cardiomyocytes, and cardiomyocyte inflammatory signaling may be enough to promote the development of an AF substrate. And finally, inhibiting inflammatory signaling prevents the development of experimental AF in various animal models. So in conclusion, for the whole presentation, despite their existence for over a century, novel conceptual models of AF are being developed and have potentially important implications for AF therapy, but require careful further study to really understand what causes and maintains AF in man. It may be possible to develop improved electrically acting anti-AF drugs. Testing on human models early in development may be critical based on the importance of species differences in ion channel expression and action potential determination. And finally, upstream therapy targeting inflammatory signaling is a promising new approach to manage and prevent AF. Thank you very much.
Thank you, Dr. Nattel, for a very comprehensive and relevant presentation. So we have time for some questions. And if you have a question for either of the speakers, please write it in the box in the lower right corner of your screen. I have one for Najah. Najah, I think there was some confusion about the source of the hearts. These are not from patients, correct? These are from consented donors, the hearts that AnaBios receives. Najah, are you on mute?
Yeah, thank you, Gary, for unmuting me. Yeah, we need to clarify that the hearts we recover, we work with organ donor centers, and the donors are dead brain donors, so we don't really recover our hearts, from patients. But donors can be normal donors, donors with normal hearts, or donors, that they have heart with atrial fibrillation.
Thank you. Another question for your presentation, Najah, was the atrial APD from AF patients did not appear to be shortened, which is one of the proposed mechanisms for AF. Any explanations for this observation?
I mean, the action potential, shortened. They showed the, a normal action potential duration, but when you record action potential, you can have action potential with normal morphology, and some of the action potential can go and become triangular or show triangular morphology. So this is an indication about the level of atrial remodeling that can take place in a donor.
Okay, thank you, Doc. Yeah, go ahead.
Yeah, I was muted. So in fact, in patients with most patients with AF, of course, it depends on the specific type of AF, because some patients have mutations in ion channels that shorten action potential duration. But certainly in the clinical series of AF patients that have been studied in the EP lab, action potential or refractory period shortening is not a ubiquitous finding. It's mainly present during ongoing AF and will remain in vitro for a certain period of time, but certainly patients who are prone to AF don't necessarily have action potential shortening or refractory period shortening.
Thank you for that clarification. Dr. Nattel, here's a question. Can drugs to treat AFib be tried on atrial arrhythmias or premature atrial beats? So in other words, treatment for atrial arrhythmia that is not AFib.
Okay, so that's a very good question, and very important to recognize in drug development, because there are different mechanisms obviously involved in AF, and it's very convenient to study antiarrhythmic drugs on patients who are known to have a high atrial ectopy burden, and to use indwelling implanted electronic devices in order to record the burden of atrial extrasystoles and look for an antiarrhythmic effect. And that statistically is very powerful and convenient and can be done in a lot of patients. The big problem with that is that the mechanism resulting in atrial ectopic activity is probably abnormal calcium handling and DADs, whereas the mechanism that maintains AF in many patients is reentrant, and any drug that targets action potential duration to try to prevent AF maintenance and reduce the burden of AF by reducing reentry is unlikely to have a major effect on the number of APCs. So you really have to be careful in designing a study that intends to look at potential anti-AF targets to know that your index of effect on AF really reflects the mechanism you're trying to target.
Interesting, thank you. Here's another question. The change in activity of different phosphodiesterase plays a role in AF. What do you think about drugs related to PDEs as promising treatment for AF?
Phosphodiesterase inhibitors, particularly. Right, so they may have value. They haven't, to my knowledge, been used. I would expect that their biggest value would be against calcium-mediated ectopic activity, which is related to the cellular calcium burden, which is enhanced by or which is modulated by phosphodiesterase. Actually, I take it back, you want to enhance phosphodiesterase activity to reduce intracellular cyclic AMP, to suppress it. So yes, there might be potential and it could be interesting, but it's basically uncharted territory. So there hasn't been, to my knowledge, a drug that's been developed yet that's effective to target the role of calcium handling in AF. That having been said, calcium handling seems to be very central in both ectopic activity generation and in atrial remodeling. So it's a target that could be a home run if you're very lucky, but the chances of being that lucky are not extremely high. I mean, that's true of any important breakthrough discovery. Yeah.
It could be something to explore with the human atrial model that Najah presented. Dr. Nattel, here's another question for you. With the variety of animal AFib models ranging from mice to human ex vivo hearts, can you comment on relative value and the relation between these different models?
Sure. So the value really depends on the target you're looking at. I think mouse models are reasonable, quite reasonable in terms of remodeling targets. So things like inflammation, specific molecular targets that leads to tissue fibrosis and arrhythmia promotion. They're also quite reasonable, I think, for an initial look at sodium channel targets and factors that determine excitability. They're not good for potassium channels because the potassium channels that determine repolarization in the mouse are very different from those in humans and large animals. Dog models and pig models could be interesting as you move towards clinical application. So I think each model that you're working with has to be really well understood in terms of the underlying mechanisms and its relationship to man. And I don't think you can say that one model is a perfect model. I think any model that you use has to be used with an understanding of its limitations. And by the way, I would point out also that when you're working with humans, clinical manifestations of AF and clinical AF syndromes are very different too. So you have to be very careful once you start bringing a drug to man to understand the differences in generation of AF according to patient age, degree of heart disease, persistence of AF, duration of AF, and all of these factors can be very important. So basically, you need to understand the animal model, need to understand the clinical model, need to understand the relationship between the two to intelligently ask questions. And then, of course, when you go to your clinical studies, you have to be very careful to select them well to minimize cost and maximize your yield in terms of finding out what the answer to the questions you need to know for the drug you're developing.
Right. Thank you. Here's another question for you, Dr. Nattel. Does endo-epi-rotor spiral wave happen in rodent atria? They are very thin.
Yeah, that's a good question. Probably, I don't know. It probably doesn't, but I don't know. Very good question. Okay. Rodent AF tends to be much more short-lasting than clinical AF, and so even in these mouse models with extensive remodeling and a lot of spontaneous AF, the individual AF episodes will still be, of course, much shorter lasting than in humans. So for sure, the details of the mechanisms are different. And again, that's why you have to look intelligently at the model that you pick and the question you're asking.
Right. Okay, here's another one for you. Is it possible to use thermal differences in inflammation as a diagnostic for AF mechanism?
That's an interesting and probing question. And so at the first level, the answer is nobody has a clue. At the second level, the inflammation that's been found in AF patients, as opposed to, for example, postoperative AF, which may be very active inflammation, is relatively low grade and local. So I would be surprised if thermal differences could be used. But the fact that I would be surprised doesn't mean it's impossible.
Right, thank you. Najah, here's a question for you. Can you comment on the level of electrical remodeling in the tissue cells you worked with, the duration of AF of these patients or the donors?
You mean the donors that the hearts that we recovered, what was a characteristic of the AF? Yeah, but actually I don't remember every case, but for every donor heart we recover, we have if we have the medical record of the donor, we can get this kind of information about the characteristic of the AF and also about the medications that the donor was taking, to control his AF or her AF.
Okay, thank you. Dr. Nattel, someone asked from the audience, would it be possible to use 3D printed human hearts as models for AF?
3D printed human hearts, you mean based on iPS cells? Is that the question?
I presume so, yes. I presume.
So the iPS cell model is very interesting and has a lot of potential to study aspects of electrophysiology. It has a lot of limitations that people need to recognize. First of all, the cellular phenotype is not that of a mature atrial or ventricular cell. People have gotten better at differentiating iPSC cardiomyocytes into atrial-like or ventricular-like cells, but they're certainly still not the same as adult human cardiomyocytes. That's a very important limitation. And in terms of using tissue preparations derived from such cells, again, there are very important limitations to date. For example, the conduction velocity in the tissue preparations that have been developed, to my knowledge, is extremely slow and not at all like that in mature human atrium. So you get conduction velocities of the order of 0.5 centimeters per second or 1 centimeter per second instead of 50 to 100 centimeters per second. And that obviously is going to have a big effect on arrhythmia occurrence and inducibility. So I think it's an interesting technique and it may improve greatly in the future, but at the moment it still has very major limitations that need to be considered in using it.
Okay, thank you, I agree. So I'd like to thank both of our speakers today for excellent presentations and a really interesting discussion afterwards. I'd like to thank my colleague Gary Watkins, Director of Marketing AnaBios, for organizing this webinar. Be on the lookout for future webinars from AnaBios. And again, thank you all for participating, and we look forward to seeing you again in the future. Thanks, and goodbye, everyone.
Thanks very much.
Thank you.